In 8.36, we need to precisely explain the light-independent reaction, which we abbreviate as ILR. But before we do that, why don't we review quick the LDR, or the light-dependent reaction. So I'm drawing part of a chloroplast here. I'm being a little bit lazy. So if you remember, in the chloroplast, there's these stacks of, I call them cookies, but these stacks of thylakoids, and their purpose is to generate a high concentration, right? Inside here, you want to create a high concentration of hydrogen protons in this area here called the thylakoid space. They will eventually diffuse out, right, through ATP synthase to make one of the products of the light-dependent reaction, which is ADP. Also, if you remember, like all over these thylakoid membranes are these proteins. And one of the jobs is to like transport the electrons to activate these pumps to pump in the hydrogen. But also, there was like one protein at the end of each one, if you want to think of it that way. They're kind of like randomly through here. But anyway, but there was one protein at the end whose job it was to take the molecule NADP and reduce it, right? Reduction is gain, to NADPH plus H. So this is another product of the LDR. So let's just review. So the LDR, that is not LDR, Mr. Fleming. So let's just review the products. So the LDR, our goals were to make ATP, which we did here, and also NADPH plus H. And those two things, which we talked about, are going to be very important in making the most important product, probably in photosynthesis, which is what we get our energy from, right? Although some might argue oxygen is important. But anyway, glucose is that main goal. And because we consume plants, or we consume organisms that consume plants, this is how we get our energy. A couple things. So we talked about this all occurring in and on the thylakoid, the production of ATP and NADPH. And these molecules will be used in this part of the chloroplast called the stroma. So that'll be a location of this whole cycle that will eventually be used to make our molecule of glucose, which we will explain in the next video.